Alrighty, so let's talk about the cell cycle and mitosis. Be ready to draw a line for your hand to get tired. Alright, so first of all, to recap, remember that we are made of 46 regular chromosomes, 23 homologous pairs, one of each comes from mom and dad. Genes are the sections of chromosomes that code for specific things. And today we're going to be talking about mitosis, which is the process of cells duplicating its DNA so one cell can split into two new cells that contain the exact same material. So before we get to mitosis, you need to understand what the cell cycle is. So as our cells are making our body work, digesting food, letting us see, making our brain send signals back and forth, cells are all going through the cell cycle, which is basically a stage that prepares them to divide. So the cell is replicating its DNA, it's going through checkpoints, it's doing all this internal stuff to be ready to split. And it does that pretty much all the time. And mitosis is the process that allows a regular body cell, so like your hair, your eyeballs, your muscles, anything like that, to split into two. So each resulting daughter cell is identical to the first one, except of course for mutations, but we'll get into all that stuff later. Now, mitosis is good for several things. If you are bacteria, for example, the only way you can reproduce is by mitosis, by splitting and making identical copies of your cells. So this is how bacteria does. But mitosis is also helpful for us multicellular creatures because it allows us to regrow from injuries. With some animals, they can regrow entire body parts. So mitosis is very helpful for us to continue to function the way we do. So we are going to draw each little part of mitosis to make sure that we all understand what it is. I'm going to pick a pretty color. What pretty color do I want? I want blue, I've decided. Okay, so we are going to draw a body cell before it is ready to split. So meaning that no DNA has been replicated yet. It is just a regular cell doing its thing. So in a regular cell, we have 46 total chromosomes. We have 23 pairs. And this is a new word. It's called sister chromatid. I'll explain what that means in a minute. So let's draw our 46 chromosomes. I'm going to draw them kind of next to each other so you can see the pairs, but I'm going to draw all 46. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. All right, so I've got 46 total chromosomes. Now you'll notice that I made each one its own individual line. That is important. So if I was just to label one chromosome, it would just be one of the lines. Now, if I wanted to label a pair of chromosomes, I would highlight the two that are next to each other. That would be a homologous pair. All right, so let's talk about sister chromatids. In the next stage, all of these chromosomes, all 46, are going to replicate. And when they replicate, they look like this. So regular chromosome, right? When they replicate, they look like this, and they're attached at the center if you were to zoom into them. Each one of these halves is one sister chromatid. So in this first stage, before anything has replicated, our sister chromatids are going to be the same as our chromosomes. Each one of these would be considered one sister chromatid. All right. So let's go on to stage 2 and replicate our DNA. All right, for this one, you're going to have to draw a lot smaller. So when our DNA duplicates, we still will have 46 total chromosomes. We still will have 23 homologous pairs. It is the sister chromatids that are duplicating. So 46 doubled is 92. So I'm going to make my pen a little bit smaller here, if I can do that. And I'm going to draw my chromosomes kind of all in the middle here, because that's what they actually do when the cell is about to split in half. So I'm going to draw 46 chromosomes along the top here. I'm going to make them look like little U's. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. All right, I told you to draw small. All right, so I have 46 chromosomes, but these are duplicated now. So I'm going to draw the duplicates on the bottom half like this. All right, so I'm going to go through. I'm not going to count them again because that's exhausting. But each chromosome is going to get its little sister chromatid friend. All right, now, just like that, we have 46 chromosomes still, 23 pairs, 92 sister chromatids. So let's label this a little bit. So again, I'll do this in a different color. Just the top half, so like just one of these little thingies would be one cis chromatid. This entire thing is still considered one chromosome. And then things still work in pairs. So any two of these would be a homologous pair. All right, so we have officially duplicated our DNA, and now we are going to split in two. All right, so when we split in two, what happens is that the top half of this cell is going to go into one cell, and then the bottom half is going to turn into another one. So it's actually the sister chromatids that are being torn in half in this process. That is how you get two new cells. So we're going to draw these pretty much the same way we drew them the first time. So after the split, each cell here will have the 46 chromosomes. They will have 23 pairs, and they will go back to having 46 chromosomes. So let's draw our 46 chromosomes in pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. All right, so we've got 46 chromosomes there, and let's draw 46 over here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. Alrighty. And same deal here. Each one of those squiggly lines is one chromosome / one sister chromatid. And then any two together would be one pair of the homologous chromosomes. All right. So I hope that makes sense. Make sure you go watch this video at the bottom here.